Okay, this is the Professor, CBMD PhD, and today's Thursday, August 3rd, 2023. It is 3:24 and 33 seconds, AM Eastern Standard Time, and today's research, cell death. So let's jump to it here. The team identifies key driver of cancer cell death pathway that activates immune cells. Date July 31st, 2023. Source: University of Illinois. The scientists have identified a protein that plays a pivotal role in the action of several emerging cancer therapies. The researchers say the discovery will likely aid efforts to fine-tune the use of immunotherapies against several challenging cancers. Most anti-cancer drugs cause cancer cells to shrivel up and die in a controlled process known as apoptosis, but apoptosis does not usually strongly activate immune cells. However, a few emerging cancer therapies cause cancer cells to swell up and burst. The protein we identified, a sodium ion channel known as CRPM4, are critical for cancer therapies that promote this type of cell death, called necrosis. Unlike apoptosis, necrosis strongly signals the immune system to target and clean up the remains of the dying cells. This suggests that treatments that promote necrosis may improve immunotherapies against solid tumors. The researchers developed two drugs, a compound called BHPI, and later a more effective agent known as ERSO, that spurred necrosis in solid tumors, dramatically shrinking and often eradicating primary and metastatic tumors in mice. These drugs work by binding to estrogen receptors on cancer cells and pushing a normally protective cellular stress response pathway into overdrive. This pathway, the anticipatory unfold protein response, or a UPR, ultimately causes the cell to swell, leak, and die. Even though they identified the initial steps, a UPR pathway that kills cancer cells, the specific proteins that mediate cell swelling, rupture, and rapid necrotic cell death remained unknown. Researchers screened breast cancer cells by knocking out each of the roughly 20,000 individual genes in the cancer cells, and then treating the altered cells with BHPI or ERSO. Cells that resisted treatments with these agents revealed which genes were essential to the drug's effectiveness. The researchers were surprised to find that TRPM4 emerged as a key driver of the process of necrosis in cancer cells treated with ERSO and BHPI. The team also found that TRPM4 was important for the activity of several other necrosis-inducing cancer therapies, and this will enable physicians to identify patients most likely to benefit from necrosis-inducing therapies because their cancers have high TRPM4 levels. The further study revealed that an ERSO-induced increase in intracellular calcium causes the TRPM4 channel to open, allowing sodium ions and water to flow into the cell. The influx causes the cell to swell, rupture, and leak, activating immune cells and causing them to rush to the site of the dead cells. As cancer treatment has been transformed by immunotherapy, which takes the brakes off immune cells, but immunotherapy has had limited success against solid tumors such as ER-positive, breast cancer, and pancreatic cancer. The scientific investigators and microbiologists say they found that the cancer drug ERSO acts like the starter on a car that turns over the engine and then is no longer needed once the engine is running. It is a swelling caused by TRPM4 that drives a lethal stretch that kills the cancer cells. As little as a 1-hour exposure to ERSO effectively killed cancer cells days later. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.